two diseases in which this type of polyglucosan bodies accumulate. One is called Lafora disease, and that is a severe epilepsy of children and is fatal. And the other, of course, is the adult polyglucosan body disease. My lab is focused on trying to understand why and how these polyglucosan bodies form and use that knowledge to try to find treatments for these patients. Now, these polyglucosan bodies are essentially starch depositions or starch-like depositions in the brains of our human patients. Now, normally, as you know, when we say polyglucosan, those are chains of glucoses, and the chains of glucoses we normally have are in a structure called glycogen. What you're seeing in front of you at the picture is how a glycogen molecule looks like. And you should note that it is extensively branched. It's not linear. And what this branching does is makes the glycogen into a sphere so that only the ends are exposed to the fluid around it, and the inside parts, which I'm trying to point with the pointer, are hidden inside the sphere. And this makes the little sphere soluble because the ends are what we call hydrophilic. They are soluble. The surfaces in the inside are hydrophobic and insoluble. This is why this sphere is able to stay soluble. Each glycogen molecule can have up to 55,000 glucoses in it, and despite that huge number of glucoses, it stays soluble. And so here are some other pictures of normal glycogen. You can see it here. You can see how it's regularly branched and structured into a sphere. You see the sphere here on the right side. And if we look with the electron microscope, you will see the glycogen molecules as these little black dots. Each of these black dots is one of those glycogen spheres, each of them containing 55,000 glucose molecules. This is the normal situation. What you're seeing here is another type of glucose chain, and that is starch. Starch, as you know, is something found in plants. The starch molecule, as you see, has very few or no branches, and therefore its surface is exposed, and therefore it is insoluble, and the starch molecules precipitate and aggregate one with the other and accumulate. So you end up with a potato cell or a rice cell or any plant cell full of this mass of starch, which we like to eat, but which is not good to have inside our brain cells. In this picture, again, here on the left side is a depiction of what starch looks like, aggregate of these unbranched or poorly branched molecules. On the right side is how the glycogen can be drawn in a cartoon with its spherical shape. At the bottom, you see the actual real thing. So here on the left, I had shown you before the small glycogen molecules. This, by the way, is in skeletal muscle, but it looks the same in brain or anywhere. So these are the glycogen molecules. Here is skeletal muscle containing a polyglucosan body, and you see how vast and big it is. It is a mass of these polyglucosans, which are, here depicted as a cartoon here in the real thing. So this is the difference between the mass of polyglucosan bodies and the normal glycogen. And the question is how these normal glycogens convert to these abnormal glycogens, which accumulate into this mass of starch if you like, or polyglucosan bodies, if you like. To understand this transformation, we need to understand how glycogen synthesis, the making of glycogen, works. And it turns out that that part is not complicated. I will explain it very briefly and simply. It is rather simple. So there's an enzyme called glycogen synthase, which adds glucose units one after the other and will keep on adding unless something happens. And that something is that a second enzyme, called glycogen branching enzyme, monitors the situation. Every time it sees that 6 units have been added, it chops it off and puts it at a branch point, connects it to another branch. So now you can see what's going on. Glycogen synthase is elongating. Branching enzyme is chopping what glycogen synthase elongated and putting it at a branch point. And the tango of activities of these two enzymes is what makes the glycogen molecule grow in a spherical fashion rather than a linear fashion. If glycogen synthase were to continue adding glucoses without branching, it would be linear and it would be starch-like and polyglucosan-like. I will show more pictures to make it more clear. So here is how glycogen synthesis normally happens. Glucoses are added, 1, 2, 3, etc. So glucoses are added, and then branching enzyme takes this and branches it. Now you have two ends, okay? It's a fork. They're elongated by glycogen synthase, and branching enzyme branches it. Now we have 4 ends. They are elongated by glycogen synthase, and branching enzyme branches it. You can see what's going on. You can see how it's growing as a sphere. Therefore, to get the normal glycogen sphere, you need balanced activities between glycogen synthase and branching enzyme. And that leads to normal structure of glycogen. If you do not have balanced activities, something like this could happen. Let us say you do not have enough branching enzyme. Glycogen synthase will elongate and will keep on elongating because there's no branching enzyme or there's insufficient branching enzyme. And so look what it's looking like now. It's no longer looking like the nice sphere. It is looking something like this, which looks like this, which precipitates. So if you do not have the balanced activities between glycogen synthase and branching enzyme, you end up with the starch-like polyglucosans and the polyglucosan bodies. Therefore, the cause of polyglucosans and polyglucosan bodies is either too much glycogen synthase activity, meaning too much elongation, or insufficient branching enzyme activity, and that means insufficient branching. Either of these will lead to the formation of polyglucosans instead of normal branched glycogen. This is an electron micrograph of a polyglucosan body, and you can see these filamentous unbranched polyglucosans in a mass in this body. As I said, there are 2 diseases with polyglucosan bodies, Lafora disease and adult polyglucosan body disease. For reasons that are not clear, in Lafora disease, the polyglucosan bodies are located around the nucleus of the cell and in dendrites. What are dendrites? You are seeing 2 neurons, 2 brain cells. The brain cells talk to each other through a connection between what's called an axon, which is this long part, and dendrite. So the axon of the neuron on the right sends a message to the dendrite of the neuron on the left through what's called a synapse. In Lafora disease, the polyglucosan bodies are in the dendrite, are on the receiving end of the message transmission, and also in this perinuclear region. In adult polyglucosan body disease, the polyglucosan bodies are in the axon or in the axon hillock here at the beginning part of the axon. Lafora disease, we believe, is due to increased glycogen synthase activity, and adult polyglucosan body disease is caused by decreased branching enzyme activity. This is a picture from adult polyglucosan body disease. These are nerve fibers taken from a patient from a biopsy. You see the long nerve fiber, and you see right inside it, there's these big polyglucosan bodies. And, you know, even a non-neurologist, non-pathologist can clearly see how these neurons will not be able to work because they're clogged by these polyglucosan accumulations. So, to generate normal glycogen, we need balanced activities between glycogen synthase and branching enzyme. In Lafora disease, we have too much glycogen synthase activity. Therefore, we thought that to fix Lafora disease, we need to reduce glycogen synthase activity back to normal, to return to the balance. In adult polyglucosan body disease, we have insufficient branching enzyme activity. Again, to fix this problem, we would need theoretically the same approach, namely decreasing glycogen synthase, in order to regain the balanced activities between the two enzymes. Now, how does glycogen synthase get activated? Glycogen synthase, the enzyme, gets activated by this other protein called PP1, which is a phosphatase. And what happens is this third protein called PTG takes PP1 onto glycogen synthase, and PP1 acts on glycogen synthase and activates it. If you don't have PTG, you cannot activate glycogen synthase. No PTG, no activation of glycogen synthase. So we reasoned that if we got rid of PTG, we might decrease the activity of glycogen synthase and stop Lafora disease. We've done this in Lafora disease so far, and in the end I'll say how we should do the same in adult polyglucosan body disease. So then we